Parathyroid hormone, or PTH, is a single-chain peptide with 84 amino acids in which the biological activity resides almost entirely in the N-terminal 35 amino acids. It is synthesized by chief cells of the parathyroid glands. The PTH gene is located on chromosome 11p15.3, transcription and translation of which results in pre-pro-PTH at the ribosomes. Post-transcriptional modification results in pro-PTH, which is transported to the Golgi apparatus, cleaved to form PTH and packaged in secretory granules. PTH secretion is regulated by plasma ionized calcium concentration via negative feedback. Increased intracellular calcium concentration leads to increased calcium binding to calcium ion sensing receptors on chief cells, which are GQ alpha subunit G protein coupled receptors. Binding of the calcium ion ligands results in a conformational change with the replacement of guanosine diphosphate for guanosine triphosphate at GQ alpha subunit, dissociating it from the alpha and gamma subunits. GQ alpha subunit activates phospholipase C, which cleaves phosphatidyl inositol biphosphate into membrane-bound diacetyl glycerol and inositol triphosphate. DAG activates protein kinase C, which phosphorylates certain proteins, initiating one cascade of intracellular processes. IP3 diffuses into the cytoplasm and binds to IP3-gated calcium channels in the endoplasmic reticulum, leading to increased intracellular calcium concentration, which triggers a second host of calcium-mediated intracellular processes. These include inhibiting fusion between PTH-containing vesicles and the cell membrane, and inhibiting nuclear PTH gene transcription. At low extracellular calcium concentrations, PTH-containing vesicles are secreted into the interstitial fluid and eventually into the plasma. Circulating intact PTH has a plasma half-life of 5 minutes. At the bone, initial and intermittent increases in PTH results in bone formation, while sustained increases in PTH results in bone reabsorption. PTH binds to PTH1 receptors on bone marrow mesenchymal stem cells, which eventually upregulates the expression of bone morphogenic protein via bone morphogenic protein receptor and a SMAD-dependent pathway. It upregulates the expression of RUNX2, inducing differentiation into osteoblasts. PTH also binds to PTH1 receptors on osteoblasts, which are GS alpha subunit G protein coupled receptors. Binding of the PTH ligand dissociates GS alpha subunit, allowing it to bind to adenyl cyclase, which converts ATP into cyclic AMP, which in turn activates cyclic AMP-dependent protein kinases, or protein kinase A. Protein kinase A phosphorylates a number of proteins, which initiate a cascade of intercellular processes, including activation of PI3K AKT mTOR pathway, which leads to the AKT-mediated phosphorylation and inactivation of BAD, a pro-apoptotic factor, by dissociating it from BCL2/BCL-XL complex. AKT-mediated phosphorylation and activation of nuclear CAMP response element binding protein, or CREB, a transcription factor, which in this setting upregulates the expression of both BMP receptor and BCL2 expression, the latter being an anti-apoptotic factor. Increased activity of hypoxia-inducible factor alpha, a transcription factor, which upregulates the expression of VEGF, promoting angiogenesis. Activation of the Notch pathway, which eventually upregulates osteoprotegerin and inhibits the activity of sclerostin. Activation of the MAPK-ERK pathway, or RAS-RAF-MEK-ERK pathway, accelerating aerobic glycolysis. Phosphorylation and activation of salt-induced kinase 2, which promotes increased concentration of dephosphorylated CRTC2, a key cofactor for CREB, and increased concentration of dephosphorylated histone deacetylase 4 and 5. These latter two inhibit nuclear monocyte enhancing factor 2C, which downregulates the expression of SOST gene, decreasing sclerostin, an inhibitor of the WNT pathway. The net effect of the WNT pathway activation promotes bone formation. Overall, the initial net effect is the increased survival, metabolic activity, and osteogenic function of osteoblasts. However, with sustained elevations in PTH and stimulation of its receptors, activation of CREB leads to upregulated expression of TNFS11, the gene encoding rank L, and downregulated expression of OPG gene. It also promotes the upregulated expression of monocyte chemotactic protein 1, which promotes differentiation of osteoclast precursors. Osteoprotegrin is a decoy receptor which competitively binds to rank L, limiting its interaction with rank on the osteoclast precursors. But with increased rank L OPG ratio, there's increased stimulation of rank by rank L, inducing differentiation and increased reabsorptive activity of osteoclasts. The bone reabsorptive effect of PTH is further supported indirectly by its influence on 125 vitamin D. In the kidney, at the proximal convoluted tubule, PTH binds to the basolateral PTH1 receptor, a GS G-protein coupled receptor, resulting in the downregulation of sodium phosphate cotransporter. This leads to decreased reuptake and increased secretion of phosphate ions. It also results in upregulation of mitochondrial 1-alpha hydroxylase, which catalyzes the hydroxylation of 25-hydroxycholecalciferol in the 1-alpha position. At the thick ascending loop of Henle, PTH binds to the basolateral PTH1 receptor, a GS G-protein coupled receptor, resulting in increased activity of the sodium-potassium 2-chloride channel and subsequent increased paracellular transport of calcium ion. At the distal convoluted tubule, PTH binds to the basolateral PTH1 receptor, a GQ G-protein coupled receptor, resulting in the upregulated expression of apical TRPV6 calcium channel and basolateral sodium-calcium antiporter, facilitating increased calcium reabsorption. Proteolytic cleavage and inactivation of PTH occurs via the hepatic Kupfner cells. For physiology, pathophysiology, or pathology, be sure to like the video, subscribe, and follow us for more.